Okay, so now we know how we get our transgene contained within the tDNA of our Bin19 plasmid in the agrobacterium into the host plant genome. So the example that I've been talking about for the last couple of videos about us trying to insert an anthocyanin gene within the genome is going to come up here. So using the binary system method, which went over last time, integration of the transgene into the host plant genome is random. So when we are infecting these leaf discs with our agrobacterium, our LBA4404 agrobacterium, with both of our plasmids with the Vir genes and our transgene in the left and right borders, when the agrobacterium infects those leaf discs, it's going to be inserting the tDNA from the Bin19 plasmid into the host plant cell genome, but we don't know exactly how many times it's going to integrate itself within the host genome. So it's kind of just up to chance. The agrobacterium may insert only one of these transgenes into the plant. It may insert it 10 times into the plant cell genome, we don't know. But we need to separate the plants that have enough of this transgene from those who don't, and we basically just do this through a screening process where we already know what we're looking at in the plants. For the case of anthocyanin, we want purple tomatoes. The deeper the purple color, the more of this anthocyanin gene expression. So we're going to select these deeper purple tomatoes for breeding, and then we have our GMO. Within the borders of the Bin19 plasmid, so in the transgenic DNA, we also have promoters and terminators, which are going to kind of regulate how much this transgene is being expressed within the plant. And because we're trying to make a GMO that's producing a lot of this transgene, we want quite powerful promoters, because a powerful promoter is going to produce lots of our gene product, which is what we want. So the promoters that we use tend to be from pathogens who have evolved these powerful promoters, because the pathogens want the exact same things we do. They want the gene they're inserting into the plant genome to be expressed as much as possible. So we steal these promoters and we put them into our we put them in front of our transgene, so our transgene is getting a lot of transcription and then translation. So we typically use the promoter's Nopalin synthase, the Nopalin synthase promoter, which comes from one of these Bin19 plasmids that we were talking about in the last few. You see the bottom one in this image, Nopalin. We're just using the promoter for this Nopalin gene, so for producing this carbon-rich sugar. And we also typically use the terminator for this Nopalin gene from that Bin19 plasmid too. Or we could also use the California mosaic virus 35S promoter, which, if you remember, was the same promoter used by Bolcombe in his experiment with the petunias, where he discovered the process of RNA interference. That's why I've got a little picture of the petunias on the bottom right there. Different host plants will have different amounts of tDNA incorporated into their genome, because incorporation by the agrobacterium is random, like I said before. So for the case of the anthocyanin gene being inserted into plants to produce GMOs, we're going to talk about these genetically modified transgenic tomatoes now. So in 2008, a scientist called Butelli expressed the genes Delia and Rosea 1 within tomatoes. They took these genes from snapdragon plants, which are these flowers shown on the right here that look a bit like foxgloves. And these Delia and Rosea 1 genes are involved in anthocyanin production. They did the binary system that we've been talking about. They inserted these Delia and Rosea 1 genes within the LAC-ZED gene within the polylinker of the tDNA in the BIN19 plasmid in between the left and right borders, and then they used triparental mating, put that in an agrobacterium. The agrobacterium containing the other plasmid in the binary system, the PAL4404 plasmid with the Vir genes, and then these Vir genes were responsible for transporting these transgenic Delia and Rosea 1 genes into the tomatoes. And then they were screened like we just talked about, so we only get the deep blue, purple tomatoes with the deep, with lots of anthocyanin in production. So these engineered tomatoes shown on the bottom right show strong anthocyanin accumulation in the peel and flesh, which you can see better on this image in the bottom right here. These anthocyanins are produced through a series of events called the phenylpropanoid pathway, which you don't really need to know. I just thought I'd include it there in case you're curious. And these anthocyanins are important to people, in humans, for our health as they're strong antioxidants, which means they pick up these things called free radicals in our body, and these free radicals are responsible for causing mutations in our DNA, and these mutations could be oncogenic, so they could be cancerous. So if we eat more of these antioxidants, we have more of them in our body picking up these free radicals, so we're getting less mutations in ourselves, and it's reducing our chance of getting cancer. A study was done on mice lacking a cancer suppressive P53 gene. These mice were fed a diet supplemented with 10% anthocyanin containing purple tomatoes that we've been talking about. And the mice lacking this P53 gene, which are normally heavily predisposed to dying from cancer, actually lived a lot longer than the control that were not fed the purple tomatoes, because they were eating these purple tomatoes. They had a lot of antioxidants in their body, so the antioxidants were picking up the free radicals, so there were less mutations happening within the mice genome, so they weren't getting as many oncogenic mutations causing their tumours, so they were living longer and not dying of cancer. What does this actually do to the purple tomatoes? Because we're thinking, well, that's great, we eat these purple tomatoes, we have more anthocyanins, we're less likely to die of cancer. Well, it's actually good for the tomatoes as well, because when we leave tomatoes out after we've picked them, eventually their peel cracks, and once their peel cracks, it's the same thing as damage done to our skin. The permeability barrier is broken and pathogens can enter the tomato cells. A pathogen called B. cinerea enters these tomato cells and triggers a hypersensitivity response, where the tomatoes start releasing free radicals to try and kill the pathogenic B. cinerea to get rid of the infection. But the B. cinerea want this because they're actually necrotrophic pathogens, so they feed on the dead tomato cells, because when the tomatoes are releasing these free radicals to kill the pathogen, they're also killing their own cells, and the B. cinerea are exploiting this to get food for themselves, and then the cycle continues, and then eventually the tomatoes just become a squishy blob, like a mess. You can't do anything with it. But because our purple tomatoes have a lot of anthocyanins, they're picking up these free radicals from this hypersensitivity response, so the B. cinerea can't exploit it. They can't get any dead tomato cells, so they essentially starve to death and die off, which is what we see in this image on the bottom left here. You can see that the infection shown in the little dot gets bigger and spreads in the red tomato, but it stays small and then stops in the purple.